Hello, everybody, and welcome to the final presentation in our Varroa Mite lecture series, Varroa Resistant Honeybees. So my name's Kelsey, and I work with the OBA Tech Transfer Program. So if you've been following along with this lecture series, you should have caught my colleagues discussing various aspects of Varroa mites. So just to recap, here's what we know so far. So we know that Varroa mites are the most damaging honeybee pest and the main factor in colony losses in Ontario. So our very first lecture was Glennis discussing how exactly Varroa mites parasitize brood and bees and the major and lasting impacts they cause in regards to colony health. Colette then covered Varroa population dynamics and demonstrated the importance of monitoring, and then along with that, how to interpret economic thresholds and figure out when you need to take action in regards to Varroa mites in your colony. And then finally, we had Dan just this past Tuesday discussing the different chemical controls available to us in Ontario, how they work, and the considerations to have in mind when deciding what to use and actually when to apply them. So if you were able to catch Dan's presentation, I hope he didn't make you feel too bad for Varroa mites, or we're going to have to get Glennis back here and talk more about PMS. So in Dan's presentation, he covered two of the three categories of Varroa control. So Dan talked about synthetic acaricides, as well as natural acaricides and organic acids. So the last category here in the green writing is cultural control methods, and that's what I'm going to be talking about today with a special focus on Varroa resistant genetic stock. So what do I mean when I say cultural control methods? What are they? And the first point I want to make here is that they're not chemical controls, nor are they replacements for chemical controls. So cultural controls or cultural management techniques, whichever term you prefer to use, they're non-chemical management practices that help control pests and diseases in your colony. So they're physical actions that you can apply to your colonies that control mite growth. You know, these methods, they're never going to decrease the number of mites in your colonies the same way a treatment will, but they can play a significant role in flattening the growth curve of Varroa population. So I think we're all familiar now with flattening the curve. So treatments will, if we remember Colette's presentation about Varroa growth, so treatments are going to decrease the amount of Varroa that are in your colony. That's not what cultural management techniques do. They're not going to ever make that curve go down. What they're going to do is stop that exponential growth from really getting out of control. So they're going to flatten the Varroa growth curve. So when you use these things in conjunction with treatments, so you use them alongside treatments, you can really form a robust and effective IPM strategy that reduces our reliance on chemical controls. So maybe, you know, it doesn't mean we don't have to use them, but it can get us to a place where we potentially reduce the frequency or even the strength of chemicals that we use in our hive. So getting into actually what these are, the first cultural control method I'm going to talk about is drone brood removal. So we know from Glennis's talk and Varroa mite biology that Varroa mites prefer to reproduce in drone brood because of the longer development time. So they spend a few more days underneath the cappings than worker bees do when drones are developing. And that allows Varroa to get an additional reproductive mite out of the equation. And they know that, so they target drone brood. Beekeepers can use this to our advantage and turn drone brood into a Varroa trap. So this works by encouraging the bees to produce a large amount of drone brood in a concentrated area in your hive. So you can accomplish this in a number of ways. A really easy way to do it is to give your hive a green drone brood frame. So these are frames, they're plastic frames that you can buy at any bee supply store, and their foundation, the cell pattern that's printed on the foundation, is a larger cell size, and that makes bees use it as drone brood. So when the bees draw it out, the queen will lay drone brood in this frame. So you can buy one of those commercially. I think they're about $5 now. That's where they're sitting. Or if you don't want to buy a plastic frame or a frame at all, then you can use a medium frame and put that into a deep box, or you can use a foundationless frame. So bees are going to fill up any empty space in the hive with drone brood. So as long as you give them a good space to do that in, they should fill that up with drone brood, as long as they're strong enough to do so. So with drone brood removal, you do have to make sure that you have a strong colony to do this. If you have a weaker colony, or they don't have enough resources, they're not going to produce any drone brood. So, no matter how often you go back and try to remove the drone brood, if there's none to remove in the first place, then this isn't going to do much for you. So this does take strong colonies to make it effective. So when you put the drone brood frame in, or allow the bees to create drone brood in an open space, you're going to let that happen until the drone brood is capped over. So until the Varroa mites crawl in that cell and get trapped by that capping, and then you're going to remove it from the colony. So you can remove drone brood by either scraping it down and putting it into a burn barrel. So don't leave it in your yard by any means. Definitely put it somewhere where you can take it away. Or if you don't want to scrape it down, because it can get pretty splattery and disgusting, you can take it away and freeze it. So make sure you're freezing it for at least 24 hours, maybe 48 hours. And that just makes sure that everything is dead inside of those frames. So if you're giving that back to your colony, you're making sure that the Varroa mites that are in there are definitely dead before you reintroduce it into your hive. So the fact sheets that you can find about drone brood removal, or when you find this methodology in textbooks, the time frame that's given is anywhere from 20 to 28 days. So you'd go back every 20 to 28 days throughout the entire season and repeat this process and remove drone brood at that interval all season long. I do want to caution you, though, against the higher end of that time frame. So we know from drone biology, drones take 24 days to emerge. So if you're leaving it for more than 24 days, you're running the risk of allowing those drones to hatch out before you come back and take it away. So that all of a sudden turns into not you controlling Varroa mites, but breeding Varroa mites and allowing them to grow even faster in your colony than they otherwise would have. That longer time frame, it's usually given to account for the bees either building comb or potentially cleaning it out if you are giving them back, frozen, a frame that you had frozen, and they have to clean it out before the queen lays there. So that's why that higher end is given. But I mean, it is possible that even the very first day, if the bees get on it quickly, it's possible that the queen starts laying that very first day. So just make sure that you stay on top of your timing when you're doing this, and try not to let it backfire on you. It's better to stay in the realm of 20 to 22 days repeat every time. So another effective way that you can control Varroa mites in terms of a cultural control method in your colony is brood breaks. So a brood break is a period of time when larvae are not being capped in the colony. So this has two benefits. One, when there's less brood, there's a higher proportion of Varroa mites physically on adult bees, and that makes them vulnerable to being knocked off or groomed off by bees, or potentially vulnerable to treatments if you're putting a treatment in your hive as well. And the other reason this is useful is because if bees are not developing, then mites aren't developing either. So they can't reproduce without capped brood, so it can slow the mite growth. Now, when we're talking about brood breaks, they can be accomplished in a number of different ways. Brood breaks will occur naturally in your hives during times of dearth or because of swarming. Of course, these are not situations that we want to encourage in our colonies as beekeepers. You know, we want to prevent swarming as much as possible. For a number of different reasons. And we never want to artificially take resources away from our hives and you know, put them through a dearth period, which is a period where they don't have enough resources to grow properly. So, you know, it's an unintended consequence of a bad situation, but certainly don't encourage those situations in your hive. A better way to cause a brood break is by splitting your colony. So a split is going to reduce the resources in a hive and slow bee production, and therefore slow mite production as well. And then when you're requeening your split, if you requeen with a queen cell, or even if you're just requeening a full-size colony, consider requeening with a queen cell. Because the queen cell, it's going to take a significant time before the queen cell hatches, goes to mate, comes back and starts laying in the colony. So it's going to be quite a long time before there's brood being produced in the colony again. And that's going to create the best sort of brood break that you can. In terms of having a mated queen, there's going to be a very small brood break when you introduce a mated queen in your colony. You know, if you introduce her in a cage, it's going to take maybe 5 days before she's out laying. So it's not really a significant amount of time that that the colony is going to be broodless for, but it is a small amount. But by all means, queen cells are more effective at this for this reason. There is there is a technique that's often talked about online that involves caging your queen to simulate a brood break. And we don't recommend using that method because it can have the unintended consequence of queen supersedure. So if you put your queen or cage your queen in one section and prevent her from walking around the colony and laying properly, the worker bees might think that she is failing, and it could cause them to start to replace her. So when we're talking about cultural control methods, you know, we're doing these things to improve the health of our bees. So we want to make sure that what we're doing isn't backfiring on us. So I would avoid that technique and stick to splitting and queen cells. Now, if you're scribbling that down, you don't have to worry about memorizing all of what I just said in terms of how to go about doing those cultural control methods. We do have fact sheets on all of these methods on our website. So at ontariobee.com/outreach/TTP. Okay, so the last cultural control method that I'm going to talk about today, and the one I'm going to spend the most time on, is genetic control. So genetic control can help your colony deal with pests and diseases in 2 ways. One, very simply, new queens are going to lay more and produce a stronger bee population. So this can help bees defend themselves against pests and help bees deal with stresses and certain disease issues a little bit better. So Varroa mite they're a little bit of a different case, but for all other pests and diseases, one of the best recommendations that we can give people is make sure your colonies are strong. And if you're noticing that your queen is maybe slowing her egg production a little bit, the colony population isn't maybe as strong as you would think it should be, then requeening with a younger queen can give your colony a bit of a boost in terms of population. But requeening with queens that have been specially bred to possess pest and disease resistant traits is going to give your bees an added advantage when handling pests and diseases. So instead of a beekeeper dealing with every single issue, you know, using queens with specific genetics is going to arm your bees with a stronger defense of their own and increase their social immunity in the hive. So it seems like a bit of a hands-off approach from the beekeeper's point of view, but making sure that your bees are able to deal with issues themselves is going to make sure that you know, it's a lot stronger and more stable of a colony. So, getting into it a little bit, I just first wanted to add a bit of context to our discussion before we get into specifics and selecting for specific traits. So, I want to take a step back and discuss natural and artificial selection in general. So, natural selection, it's the process of a population changing and adapting to their environment. So, this change is going to occur when an individual in a population has some sort of competitive advantage over other individuals in a population. This advantage increases their chance of survival and increases their chance of reproduction. So it increases the chances that the trait is going to be passed on to the next generation. This happens when this happens in enough individuals, the next generation is going to have higher levels of this advantageous trait. And slowly, if the trait in question remains to be advantageous, then the population is going to progress in that direction of that advantageous trait. So an example here, a classic example of natural selection is the peppered moth example. So in peppered moth populations, there's natural variability in their coloring. So moths can be light colored or dark colored, and this trait is heritable. So it's passed on from one generation to the next. Light colored moths have light colored babies, dark colored moths have dark colored babies. Before the Industrial Revolution, the light-colored moths were the most common phenotype observed, and dark-colored moths were quite rare. So this could be explained because the light-colored moths could easily camouflage themselves on light-colored trees, whereas the dark-colored moths you can see in this photo, they stand out, which makes them more vulnerable to predators. During the Industrial Revolution, coal-burning factories spewed soot and pollution and darkened trees and darkened forests. This made it easier. It sort of flipped the tables on them. So all of a sudden now dark colored moths were able to better camouflage on the tree, and light colored moths stood out and started getting eaten by predators more. So the dark colored moths who survived, who didn't get eaten, were able to reproduce and pass on their coloration to the next generation. Eventually, dark-colored moths emerged as the dominant phenotype and were more commonly observed than their lighter counterpart. So the competitive advantage of color here and camouflage in their environment allowed them to survive, reproduce, and pass on their heritable traits onto the next generation at higher rates, and it shifted the average phenotype of the population. So you can see that there in the graph on the right-hand side. So early on, the light-colored moth was more common. And then as time went on, as conditions changed and dark colored moths started to have a competitive advantage in that environment, you can see selection change the direction of the average phenotype of all of the peppered moths populations. So when it comes to breeding, breeding is artificial selection, basically. So instead of it happening naturally, you're changing the conditions and you're choosing the traits that get passed on. So it's artificial selection. And this has been happening for centuries. Even before formal breeding programs were established, there was inadvertent selection that occurred simply through the domestication of animals. So when you change the environment of a species, natural selection is going to occur. Farmers who kept animals caught on to the phenomenon of heritability and started selecting their preferred individuals to reproduce and pass on their genetics. So in this deliberate system, trait selection can occur much more quickly than it would otherwise, or depending on the trait being selected, than it would at all. So this might be a little difficult to see. I know the writing is tiny here, but this is just a table of showing some of the earliest domesticated animals. And you can see here's where our honeybees land. So they were pretty early in terms of domestication. So this is just domestication. When we talk about formal breeding programs, they were first established for the more economically significant livestock. So talking about sheep and horses, especially cattle, those were all quite early. And now, of course, breeding programs exist for just about everything. That being said, you can't just breed out of nowhere. There are some specific requirements that you need to have in place for successful selective breeding. So the traits that that are being considered and that you would like to select for, they need to be measurable and heritable. So that is to say that there must be a way to quantify and rank the trait in question, and it has to be a trait that will be passed on genetically from one generation to the next. Early breeding programs made use of subjective observations about the best performing individuals just based on the farmer's preference. But now a truly successful and robust breeding program that's going to make the most progress possible in terms of your goal and towards your goal, it needs to employ objective and standardized evaluations to rank individuals. So just being very clear about what you're looking for and developing standardized tests to be able to compare individuals is critical for a breeding program. And then to that point, there also needs to be variation in individuals to be able to rank them at all and make meaningful selections. So we need a degree of genetic diversity. You can imagine going back to the peppered moth example, if all of those moths didn't have variation in their coloring to begin with, if they were all purple moths, for example, there'd be no way to select for one over the other, one individual over the other. So you do need a degree of genetic diversity. And then selections need to occur in a way that you're not sacrificing genetic diversity. You're not making selections that are so narrow that the genetic diversity of the individual is negatively impacted. This is something we have to keep in mind for honeybees, especially because at a certain point, genetic diversity it affects the health of the overall organism. So it does have to be present there. So you want to make sure that you're not making negative consequences based on the selections that you're doing. It's also important for breeders to have a specific goal in mind for what they're after. And they have to know that the goal is going to be reached by future generations. Selective breeding doesn't result in perfection after one single generation. Selections have to be continuous and consistent and ensure progress is made in each successive generation for a very long time. And each generation making small steps towards a larger goal. And that goal is going to happen far down the line. When it comes to breeding programs for honeybees in particular, all of the requirements I just talked about apply. One thing to keep in mind, when I mention individuals for honeybees, you're not making selections necessarily on individual bees or there are some selections that happen on the queen as an individual, but for the most part, when we're talking about selecting honeybees, it's on colonies, not individual bees. So you're making your selections based on the overall colony performance. And there are many breeding programs that exist for honeybees. Some of them are focused on overall performance and creating a very practical bee that's well balanced and suitable for use in a commercial or hobbyist operation. And then there's some that are bred for just one specific trait, and those are usually in research situations and for research purposes. For honeybee breeding, in addition to the other requirements, there are some additional requirements that you need to keep in mind, or not requirements necessarily, but things to consider when you're talking about bee breeding. So honeybee breeding is complicated. In many ways, it's a lot more complicated than other livestock breeding, and that's because it can be quite difficult to control. Queen's mate in flight, far from her source colony, with up to 20 drones. So honeybees are polyandrous. So she mates with many, many drones, and she keeps sperm from all of them. So that's a lot of genetic input that goes into the next generation. So those are both physical and genetic complexities to contend with there. And I'm not here to discuss those complexities today. So this presentation is really going to be focusing on specific traits that can be bred for, rather than how to set up a successful breeding program overall. But if you'd like to learn more about breeding honeybees in particular, there are some great resources out there you can check out. These are some of the ones that Tech Transfer picked out as definitely needing to be looked at if you're talking about bee breeding or learning about bee breeding. But these certainly aren't the only ones. So by all means, get out there and start learning. But for now, we will keep it narrowed down. There's just way too much material to cover already. Forget about getting into developing breeding programs for bees. So the importance of breeding honeybees, why is it important? And by all means, breeding bees is not going to be for everyone. I think we're all drawn to and appreciate different aspects of our beekeeping industry. So we have honey producers, we have pollinators, hobbyists, in addition to queen breeders. But no matter what we do personally, we should appreciate all of the different sectors and the importance of each one. There's not one that's more important than another. We do need all of these pieces to come together to form our industry. And breeding bees, even if you're not a breeder yourself, it's important to understand and appreciate. So breeding can lead to a bee that thrives in our local environment and increases the sustainability of our industry. So bee populations can be bred to possess traits that make them beneficial to the beekeeper, no matter what their goals are. Things like overwintering ability, disease resistance traits, those are things that are good no matter what you do with your bees, and they're things that are good for a bee population in an industry to possess. And then bee populations can also be tailored to fit in with specific goals of the beekeeper. So you can select for traits like high honey production, strong spring buildup, low defensive behavior, things like that. So depending on the goals of the beekeeper, you can really sort of design a bee that is everything you want it to be, or close to it anyway. Remember, breeding does take work, and it's not perfection all of a sudden. And all of these things that we can do, they increase not just the chances of our bees survival and our bees success, but the success of the beekeeper as well. And why breed for Varroa resistance in particular? So when it comes to Varroa resistance traits, why is breeding important? If we already have the other controls like chemical controls and the other cultural management techniques that I talked about earlier today, why is it worth the effort of 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 creating a breeding program and incorporating these traits into a breeding program? And I think to answer this, we really need to keep our principles of integrated pest management in mind. So depending on a single solution, even if it's, or especially if it's a chemical, that can be risky. It's risky for a beekeeper, and it's risky for an industry to do something like that. I've used the word resistance a lot tonight, and I'm going to use it more, and that's largely in a positive way, talking about Varroa resistance. But resistance can also be a bad thing, because Varroa mites can and have been known to become resistant to chemicals. So if we're able to decrease our reliance on chemical controls and get to a point where we have to put them on our hives less frequently, or at lower strengths, that's going to make sure that the chemicals that we do have remain effective long term. So when we do absolutely need them, we can reliably put them on our hive and know that they're going to knock down those levels. It's all about using all of the tools in your tool belt to form a dynamic IPM plan. And that's ultimately going to lead to the sustainability of our industry. Now, I smile when I say that because I think every single presentation so far from all of my colleagues and myself now, we have used the phrase, use all of the tools in your IPM tool belt. And that really is something critical that we need to get a handle on and understand as beekeepers. So by all means, it's not the last time you're going to hear that. Getting into the actual mechanisms of resistance. So what are they? What are these behaviors that allow colonies to defend themselves against Varroa mites? And there may be many different mechanisms that confer Varroa resistance in bees. Some that we don't know about, some potentially that we don't fully understand. I think there's a lot of room for research in this area. So I'm going to talk about a few that we know and have identified already. But by all means, there might be others out there, and there's definitely always room for research in this area. So the traits that are listed here, they're all mechanisms that have been identified through research that confer either pest or disease resistance, and they are heritable and can be selected for. So of course, that's one of the requirements of a breeding program. The traits that we identify, we have to make sure that we can, target them and that they're passed on to future generations. So the first trait in this list here is hygienic behavior. So this is something that was first noticed in bee colonies that did not succumb to American foul brood infections. So hygienic behavior is resistance to brood diseases more than Varroa necessarily. And then the other two listed here are specific to Varroa. So there's Varroa Sensitive Hygiene and grooming behavior. So these are traits, they have been identified in other bee species. And we've found them in Apis mellifera, so our honeybee, at lower degrees, but they still have been present. So let us start with hygienic behavior. So hygienic behavior is a defense mechanism to brood diseases. And this mechanism involves two things. So first, the ability for bees to detect that something is going wrong with the brood. And second, it's their tendency to quickly remove the diseased brood. And notice I say quickly, so they do have to remove the diseased brood before it's able to spread throughout the colony. So all bees have, the tendency to tidy up their hives eventually, but making sure that bees can do this quickly, that is what's going to confer the disease resistance, because they're going to be able to get that out before that pest becomes infectious and spreads throughout the brood area. So there are a few ways to identify hygienic behavior in honeybees. If we're talking about truly selecting for disease resistance or disease immunity, it would be necessary to expose bees to a specific pathogen to test if they're truly resistant to that pathogen in particular, and then of course record their response. Now, there are biosecurity issues that go along with that. We do not want to be introducing American foul brood, for example, into our hives and causing outbreaks for the sake of breeding. Not to mention the animal husbandry considerations that go along with this. So that's something we want to avoid. And instead of doing that, we have field assays that we can do in place of testing against a true pathogen. And that simulates an issue without actually having to to weaken and sicken your hives. So there's a couple different methods that you can do for hygienic behavior, but the most effective one, and the one that's been most reliably associated with hygienic behavior, is called the freeze kill brood assay. So through this test, you freeze a section of brood and evaluate how much the bees clean out after a certain period of time, and you can give them a percent hygienic level. Colonies that rank consistently above 95% after this test are considered hygienic. Of course, they rank below that at different steps, but above 95% is group 1 or highly hygienic. For breeding purposes, you don't have to have colonies that are consistently ranking above 95%. Progress can be made simply by selecting the top colonies. So rank your colonies in regards to hygienic behavior, and then selecting from the top ones are going to move you towards hygienic behavior. So just a reminder, it's all about those baby steps. So I do have a video here to show you hygienic behavior. So this tech transfer does a lot of hygienic behavior testing in the summer. We do this for members of the ORBS program. So the first step is identifying the correct age of brood. So it does require a specific age of brood to make sure that they succumb to freezing, but they don't inflate the results. So we have to uncap cells and target that. And then we push down collars to isolate the liquid nitrogen that we pour into a specific area. And then we use liquid nitrogen to freeze that area. You can cut out a section and freeze it in your freezer, but when we're doing this in the field, liquid nitrogen is the easiest and fastest way to go about it. After that, that frame has been tested, it is returned to the colony for 24 hours and left alone. And the colony should be left alone for those 24-hour period, and then we come back and check results. So the first frame here, it's exactly what you want to see. Everything is taken out of those cells. And then this picture here is a terribly scored, it's a colony that scored terribly in terms of hygienic behavior. They've barely touched that brood. Hygienic behavior with the freeze kill assay, the freezing is, I mentioned it was the best way, but I think I should explain why it's the best way. So if you remember, it does take 2 traits to confer hygienic behavior. So the detection and the removal of that diseased brood. So the detecting part is why this one is so useful. Liquid nitrogen or sticking your frame in a freezer, it kills the brood underneath those cappings, but doesn't actually damage the cappings. So it's still testing the bee's ability to detect that an issue is happening through that capping. If there's the pinprick method where you pin prick into the cell and kill the larvae that way. But that damages the capping. So it's sort of, giving your bees a little bit of a cheat. The capping's already open, so it's easier for them to detect that something's wrong. So hygienic behavior, it is very effective as a defense mechanism towards brood diseases, but its ability to confer resistance to Varroa mites is a little less sure. So when you have high levels of Varroa infestation, studies have shown that hygienic behavior will kick in and start removing brood that's affected by Varroa mites. However, the hygienic bees usually target cells that have more than one founder's mite. So they're probably responding to the damage that's being done to that pupa rather than the Varroa mite itself. And there's another behavior called Varroa Sensitive Hygiene. And colonies that have this trait, they're more sensitive to the actual Varroa themselves. So this behavior kicks in at lower Varroa infestation levels. So this is really important because sure, it's great that hygienic behavior will kick in at higher infestation levels, but that doesn't really do much when we talk about controlling Varroa mites because that issue is already out of control. So we want to target things that allow the bees to reduce the mite load before that damage is happening. So this is really where the next couple traits come into play. So Varroa sensitive hygiene, it's specific to Varroa mites. So it's when workers uncap and remove pupa infested with fertile reproductive mites. So remember that fertile bit because that is important for the next one that I'll talk about. So testing for VSH, it can be pretty straightforward, although it does take quite a bit of time and resources to perform the test. So to start, you're going to need a significant source, excuse me, of Varroa mites. And by significant, you'll notice these numbers here. I mean, you need a colony with 15 to 35% infestation. And that's, midsummer when you would be doing this test. That is a heck of a lot of Varroa mites. If anyone has seen an infestation level this significant, it does mean basically the death of this colony. So, I mean, that's hurdle number one. So keep that in mind if you do want to test for VSH. Um, but that is how it starts. So once you have this Varroa colony, you leave a frame in it until the queen lays brood, and you remove it just after it's capped. So you do want to be removing capped brood to make sure that the Varroa mites had a chance to go into the cells and get stuck there effectively. When you take out that frame, you're going to uncap 50 to 100 cells per side of the frame to assess the initial Varroa infestation level. So you uncap the cell, you check if there's a Varroa mite in it and how many Varroa mites, if there's potentially more than one foundress. And from that, you can get a percent infestation level. You're then going to take that frame and place it in the colony that you want to test for hygienic or for VSH and leave it there for 1 week. So you're leaving it in for 7 days. After that 7-day period, you're going to come back and basically count the Varroa infestation again. So you're going to count and uncap 50 to 100 cells per side of the frame again and assess the percent of cells that are infested with Varroa mites. And then you can get the percent VSH of a colony by determining the difference between those two levels. So here are a few photos here, just showing you sort of that whole process. I had the pleasure of working with the research team at the Animal Research Center of Deschambault in Quebec a few years ago when TTP collaborated with Pierre Giovinazzo and the University of Laval on a research project aimed at breeding VSH in bees. So this is a few photos of the initial assessment. That's what this was. So you can see us removing those frames from these Varroa colonies. And I do want to make the point that they are a research center and had an isolated yard for this Varroa work. They did understand what infestation levels like that would mean. So they made sure that there were no poor beekeepers around them getting decimated by their high Varroa levels. You can see we're set up there in a tent to uncap and pull out larvae and then count the Varroa mites that come out along with them. So another trait that is very closely related to VSH, but it is a little bit different, is something called suppression of mite reproduction. So in any Varroa population, there is a natural rate of infertility of about 5 to 20%. And then as bees preferentially target and remove infested pupa with reproductive mites through VSH behavior, the percentage of infertile mites of the colony increases. So research has shown, looking at this, that VSH is one of the main factors contributing to the increase in the proportion of infertile mites in the colony because of the VSH bees, excuse me, that pull out reproductive mites. But there also is another mechanism going on there that actually suppresses the mite reproduction from the inside. So it's the larvae has some effect on suppression of mite reproduction as well. So SMR, is, as it's called, is quantified by taking a closer look inside those inside cells populated with a single foundress mite. So it is important if you have a cell that had 2 foundress mites, you can't tell who laid what in terms of the foundress and the families that belong to them. So this does require cells with only a single foundress mite. And this is done during the second VSH analysis so that after the 7-day period, when you remove that frame and do the second VSH count, this is when you would do the SMR analysis as well. Whenever you find a single foundress, you take a deeper look into that cell and dig into the Varroa family that exists inside of that cell. So based on the age of of the pupa that's in that cell, we know where the Varroa family should be. We know how many daughters she should have. We know what life stages they should be at. We know that there should be a male in there. So you can assess that and compare that to what's actually there. And if at any point it's less than what we would expect, so if there are less offspring than we would expect, if there's no male in the cell, potentially if there's no offspring at all, we know that that mite's reproduction has been suppressed in some way. Now, recapping activity is another mechanism that's closely related to both hygienic behaviors as well as SMR. So recapping activity, it's the tendency of bees to uncap and recap cells. And there does need to be a little bit more research done into this behavior, but it's thought that this recapping activity could be causing some infertility in mites. Whether the cell is uncapped and the male mite is damaged through that process, or maybe there's something about the bee opening up that cell that really ruins the mood for the brother and sister Varroa mites. But there is something there that could be causing suppression of mite reproduction. And this has, it's been used as an indicator for Varroa resistance. So you can check recapping activity by carefully removing the cell capping. So cutting around the cell and flipping that over so that the cell capping is intact when you remove it. And then you can look on the underside of it, and there should be a silk lining for when the bee shed its cocoon. If there is a hole in that silk lining, then you know that the cell has been recapped. A hole was dug into the wax and the silk lining, and then recapped by just plain wax. That silk lining can't be repaired once it's damaged. So that's VSH and the bundle of VSH mechanisms that go along with it. And now I'm going to move on to another major mechanism of Varroa resistance in honeybees, and that's grooming behavior. So grooming behavior, it's very much how it sounds. It's the tendency of bees to groom themselves or groom each other. So there's auto grooming or allo grooming. And they physically knock mites off their bodies. And in some cases, they'll even knock mites off and damage them. They'll bite them or damage their legs before they drop them down to the bottom board, so that the mites can't climb back up and reinfest another cell or climb back onto another bee. So grooming behavior, it's assessed in two ways. So first, it can be observed, or bees can be observed for grooming behavior in the lab by assessing response time as well as intensity of grooming. So the length of time that they groom themselves and the number of legs that they use when they're grooming themselves, those both go into the intensity of grooming behavior. So this is done, if you're doing it in a lab, you can have a little petri dish and add a mite onto it, and observe the bee's response after being exposed to the mite or Varroa mite themselves. It can be a little tricky sometimes to get live mites in the lab. So on occasion, there has been substitute irritants like wheat flour placed onto bees, and the grooming behavior recorded based on it being exposed to a substitute irritant. The other way that you can assess grooming behavior is by collecting sticky boards. So doing just a standard mite drop and then taking a closer look at the mites that drop onto sticky boards and seeing if they're damaged or not. Of course, sometimes it can be tricky to tell if it was actually damaged by a bee or just, based on it falling down to the bottom board or being damaged by a bee, taking it out of a cell. So, it's a little complicated that way, but it has been shown that you can tell grooming behavior based on an increased proportion of damage to mites on sticky boards. So I have a video here of a mite exposed to wheat flour. So that should be if you don't hear sound, that's okay. There is no sound associated with this video. But that that's a little bee there exposed to wheat flour. So these videos were supplied by Dr. Nuria Morfin from the University of Guelph, where they did quite a lot of research into grooming behavior. So that one, a quick little video showing them observing the bees, grooming themselves, the number of legs, the length of time, etc. Now, as you can imagine, all of these mechanisms are quite tedious to check for. So even something relatively simple like recapping activity, it takes time, it takes skill, it takes tools to assess properly. Not to mention the Varroa colonies you need for VSH and the time it takes to do grooming assessments. So it has been a pretty major barrier for incorporating any of these traits into honeybee selection programs outside of research labs that have access to these resources. Luckily, though, there has been progress in developing a very practical field test that can be used to select for Varroa resistance. So this is called low Varroa growth, and quite simply, it's a test that compares the relative Varroa growth between colonies so that breeders can select for colonies that maintain low levels of mite growth. So it works by taking a sticky board at the start of a season. In the middle, all factors remaining the same, so you're not affecting Varroa growth in any ways. You're not applying treatments or splitting excessively throughout the season. And at the end, you take another sticky board or Varroa level in it using another monitoring method. And you can compare the rates of growth per colony and then rank your colonies based on that. So the work that went into developing LVG, as we call it, it really came out of a lot of work done by the University of Guelph Honeybee Research Center. So Dr. Ernesto Guzman and the research team there, they've done extensive research into grooming behavior. And through these projects, they noticed that Varroa growth rates correlated with Varroa resistance and grooming behavior. So from those observations, they developed this project looking at LVG specifically, so really developing the protocol for this field test and seeing if this is something that can be selected for and bred for. So can it be reliably passed on from one generation to the next using this field test? So Tech Transfer collaborated with them on this low Varroa growth project a number of years ago. So basically what went into this project is that colonies were, a large number of colonies were monitored for Varroa growth. So we took a sticky board in the spring, took a sticky board 15 weeks later in the fall, and assessed the Varroa growth. So remember those Varroa growth curves, we compared each one. We selected the top, so the top 20 were the 20 with the highest amount of Varroa growth, and then we took 20 with the lowest amount of Varroa growth, so both the steepest curves and the flattest curves. And then we looked at those 40 colonies a little more extensively for things like hygienic behavior. We looked at low defensive behavior, damage to mites on sticky boards, mite reproduction, and that sort of thing. And then from that, we chose the top 3 with high mite growth and 3 with low mite growth and grafted from those, produced 100 high mite growth queens, 100 low mite growth queens, introduced those into colonies and repeated the entire experiment for 2 more seasons. So each season we grafted again to track the progress from one generation to the next. And they were able to show that you can reliably select for LVG and reliably select for low Varroa growth using this method. And the really great thing about this field test in particular, of course, it sort of comes out of being associated highly with grooming behavior, but it also selects for all of the mechanisms. So you're never going to do an LVG test and identify the exact mechanism that's causing the low Varroa growth, but it has been shown to be correlated with grooming behavior. And work that's come out of Pierre Giovinazzo's lab at the University of Laval, they have correlated it with Varroa Sensitive Hygiene. And then it makes sense, right? There might be a lot of different mechanisms causing low Varroa growth. You know, in some cases, in research cases, and for some breeders, it matters what that mechanism is. But for the practical, use of for the industry, it doesn't really matter what the exact mechanism is. It just matters that every year your honeybees are getting a little bit more resistant to Varroa mites. So really fantastic that we have this practical field assay for Varroa resistance. All right. Not to throw a damper on this parade that we've been having, because breeding really is great. But there are a few caveats I want to cover before I send you guys home. Or, I mean, I guess you are home, but I think you understand what I mean. So first, breeding for Varroa mite resistance, it does not mean you're never going to have to use treatments. If it were that easy, we wouldn't be here still talking about Varroa mites. We can't dismiss what we learned in the previous talks. So no matter what, the most important thing you can do for your mite levels is monitor and respond with chemical controls when they start getting to threshold levels. We can make significant progress as an industry towards a bee population that doesn't require regular chemical treatments. That is true. That's a future goal, but we're not there yet. Remember that breeding is slow, and total Varroa resistance has to be our goal for future generations. It's not happening tomorrow. So just keep that in mind. And then in addition, breeding's not as simple as, hands off, quit your treatments and see what survives. Breeding programs have to be robust and consistent and have predetermined goals, and a selection plan that incorporates standardized tests, not just what survives. I think sometimes we hear stories of beekeepers who go maybe treatment free, thinking the best will survive and good riddance to the rest of them, sort of applying the survival of the fittest mentality. And I can't stress enough how wrong that is. That will not and does not result in any sort of meaningful progress towards pest and disease resistance. In fact, it can come along with a lot of major negative consequences in terms of spreading pests and diseases. We live in and operate in an industry where we're on top of each other with our colonies, and our actions impact our neighbors. So even if it were a legitimate way to breed, even if you can just let your pest and disease levels go crazy and then breed from their survivals, it would still be a net negative, because those disease issues that you're letting run rampant are going to spread to other beekeepers in your area. So that's not what we want. And to be honest, that's just from a breeding and biosecurity point of view. There's also, of course, animal husbandry. We're responsible for these critters. We're responsible for making sure they are healthy and lead good lives. If we remember from Glennis's presentation on last Tuesday, Varroa mites cause a lot of damage when they get above threshold levels. And they're not the only things that are going to kill your bees either. So taking a hands-off approach, it's not going to lead to stronger stock. It's just going to make your bees die slow, horrible deaths, and not going to be a big benefit to the industry. That being said, never fear. There are people out there, there are breeders out there in Ontario who understand the difference between putting selection pressure on their hives and not causing harm. When we talk about ranking your colonies, of course, you're going to have some on, the bottom of that ranking list. You don't have to just give up on those ones. You can requeen those colonies and use them and make sure that, they're as healthy as they can be. You don't have to give up on them. And we are very fortunate in Ontario to have the ORBS, so the Ontario Resistant Honeybee Selection Program. So breeding for pest and disease resistant bees are exactly what these breeders do. So this program was started in the early 90s in the face of the threat of tracheal mites in our industry, and breeders started breeding for tracheal mite resistance. Since then, we've really sort of shifted the program to focus on hygienic behavior. So I mentioned that tech transfer does a lot of hygienic testing, and it is for ORBS breeders that we offer this to. So they really put a strong focus on hygienic behavior, as well as some other critical traits that they select for depending on their personal goals. Not every ORBS breeder has the same goals overall. Some will prefer honey production over low defensive behavior. Some will prefer spring buildup, so there's diversity and variability there, but all of them do put an emphasis on disease resistance. And in the last couple of years, since this LVG came out, we have been incorporating LVG selections into this breeding program. So there are some breeders who already select for various Varroa resistance mechanisms. So there are some breeders who've been looking into selecting for VSH or selecting specifically for grooming behavior. But this LVG, we're looking at making it just as important as a selection as hygienic behavior, and making sure that it's standard across the program. So if you get any ORBS breeders queens, you're getting those pest and disease resistant genetics in your bees. If you are interested in purchasing queens from an ORBS breeder, you can find information at orbs.ca. So by all means, go on there. You'll find a list of ORBS breeders. If you contact everybody on that list right now and everybody is sold out, check back at the very beginning of April. We are going to be updating that list and adding a couple extra breeders who just joined the program. So if you're not a bee breeder yourself, you still can incorporate genetics into your operation. You don't have to breed yourself. You just have to buy from breeders who do select for these traits. And then if you are a breeder, or if you're interested in becoming a breeder, by all means, reach out to Tech Transfer or visit the ORBS website for more information. And we're always looking to grow the program. All right, so that is all that I have for you today. So that concludes both my presentation and our Varroa lecture series overall. So I'd like to say thank you so much to everybody who tuned in. It's really great to see such a strong response from beekeepers looking to learn and maintain the health of their bees at Tech Transfer. We definitely appreciate you listening. I would also like to give a final shout out to OMAFRA, who provided funding and allowed these talks to happen.